Hey everyone. So today, for the last presentation of all, congratulations on making it this far. I'm going to be presenting on taste, flavor, and additives. So first of all, I'm really sorry I can't be there in person. I was excited to get this topic and looking forward to presenting it, but sometimes, we can't control the things that happen. But hopefully, we're still going to be able to learn lots of cool things today. So with that in mind, let's get started. The basics. What is taste? I'm glad you asked because taste is a chemical sense that humans evolved to be able to analyze different substances. Because early in our evolutionary history, when we were on the savannah, like hunting for food, if you bit into a fruit and it was not ripe, you had to be able to recognize that immediately and spit it out. Because if you swallowed it, you'd get sick and then you'd be an easy target for predators. So it was that type of survival situation that necessitated that we have a sense of taste. So, for example, that bitter and sour tastes tend to indicate either rotting meat or most plant poisons, which are also bitter. Sweet tastes, on the other hand, are great. If you run into something sweet tasting on the savannah, then you are in luck because that indicates that there's a lot of monosaccharides and disaccharides usually that can be obviously great sources of energy for you. And then lastly, umami tastes, which we would call savory in America. Umami is just the Japanese word for savory. They indicate something rich in certain proteins, so a fresh kale would taste like umami, and that's also very good. Taste is also a really interesting special sense because of how closely connected it is to our autonomic nervous system. So taste doesn't just go to the cerebral cortex; it also actually goes to the brainstem, and from there are a lot of automatic reflexes that are involved with taste. So two of the simplest are the salivation reflex that comes from pleasurable tastes, and this is also linked to it's not just your mouth salivating, but it's also your stomach and intestines starting to produce fluid. And the other one that what everyone knows is a gag reflex when you taste or smell something that's unpleasant. So let's break it down. We know there are five basic tastes, but what are they really? Sweet is the biggest one, and it's almost always activated by sugar. Occasionally, there are certain alcohols and certain sugar substitutes that we'll get into later that can bind these same receptors. But for the most part, if you taste something sweet, it could be a sugar of any type. It could be glucose, sucrose, lactose, whatever. Sour taste is probably the simplest because this is always triggered by the presence of hydrogen ions. So whenever you're having something acidic, usually fruits, you're going to have hydrogen ions, and that automatically triggers a sour taste. The more hydrogen, the more sour. Salty is these are the mineral ions that are stimulating salty receptors. So usually sodium, sometimes potassium and magnesium can also play a role in that. Most of the foods we eat, though, that have potassium and magnesium don't really have them in a high enough concentration to make it salty. So a banana, for example, it has potassium, like a significant amount, but the sugar in the banana completely outweighs that, so it doesn't taste salty when you eat it. Although the potassium is technically activating those receptors. Umami is these receptors are activated specifically by glutamic and aspartic acid, which are two of the more common amino acids in meat, fish, etc. There is glutamic and aspartic acid present in the proteins in beans and those protein-rich vegetables, but they tend to have less of these two. So those will still taste umami, but not to the same degree. And then finally, bitter taste is the most complicated because there isn't like one pattern that defines all the foods that taste bitter. Some of them are plant poisons, and some of them are entirely different molecules. Uh, and because there was such a big variety of things we needed to stay away from, bitter receptors, we have a big variety of bitter receptors in order to be able to be activated by those. So we've already discovered about 40 distinct proteins that will send a bitter signal, and we're finding more and more each year. So other flavors besides those five, there have been propositions that fatty is a flavor, and we have found a receptor that we think responds to linoleic acid, which is an acid that you would find in most of your triglycerides and your plant oils. There we go. But it's not usually counted with the others, but there are scientists today that are trying to make it the sixth taste. Hot and spicy is the other flavor a lot of people's minds jump to, but this is absolutely not a taste. It's a common misconception, but hot and spicy isn't the activation of taste bud receptors at all. It's the activation of pain receptors, which are responding to capsaicin, which is the chemical in peppers that produces this burning sensation. And yes, I do feel really bad for peppers because, all these animals were eating them. So they decided, oh, you know what? Let's evolve some really nasty chemical that'll burn their faces so that they'll stay away from us. And then humans tried it and we're like, oh, this is amazing. But, well, we got them in the end. Speaking of misconceptions, by the way, let's get this cleared up. The human tongue is not divided into regions that are each responsible for sensing different flavors. So the papilla across your tongue have taste buds for all the different flavors. You can taste sweet anywhere. You can taste sour anywhere. It is true that the big papilla in the very back, they have more bitter receptors, and that's sort of like a last line of defense because beyond the back of the tongue, if any food gets there and then we realize that there's something wrong with it, then that's our last chance to spit it out before it gets swallowed. So now we get the basics of, what is taste, what is flavor. Let's get into the actual mechanism of this. So a papilla is the rounded mass on the surface of the tongue that you see in the mirror with your naked eye. And this isn't the functional unit of taste. The functional unit is a taste bud, which are these little clusters of cells that you find on the papilla, kind of onion shaped. But in this cluster, each of these cells has the fluid around it that the chemicals of the food will enter. And they'll sort of like dissolve through here. And then on the surface of these cells, you have the little protein receptors. So you have a receptor for H+ ions that's going to activate sour taste, etc., for example. So after these cells respond to something that they receive from up here, they release ions down here that will change the polarity and cause an active potential to travel down this afferent nerve. So it's one nerve or one neuron, really, per taste bud, which is why this is like the smallest possible unit and the most specific. So obviously you send an action potential. There's a depolarization period in the neuron that it goes through. And meanwhile, the signal itself travels up the neuron until it combines with either the facial, glossopharyngeal, or vagus cranial nerves. Those are the three sensory nerves, or they're partially sensory, partially motor, but those are the three nerves that service the tongue. And those will take it to two places, actually. They will take it to the gustatory cortex of the brain, which is in the frontal lobe, but they'll also take the signal, like I mentioned before, to the brain stem in case we need to have one of those reflexes. And then once it's in the brain, we can assign the taste labels like sweet or salty based on what receptor was activated, and then we can figure out what it is more specifically, like whether it's cheese or whether it's banana, based on the olfactory information that we're receiving. So identifying the specific flavor is more reliant on the sense of smell. So let's look at the beginning of synthetic flavor now. The first modern chemical flavor additive was vanillin, discovered in 1874. And what happened was these German scientists were trying to figure a way to isolate the actual compound in vanilla, and they did this by evaporating vanilla extract until they just had like these little solids remaining, and they recrystallized those solids. And based on its crystalline form, they were able to deduce its molecular form. And they found, wait a minute, this is something that we can make ourselves. We don't need plants to make it for us. So with that synthetic flavor really took off because the production of vanillin turned out to be something incredibly competitive. Vanilla beans were a rare commodity, and they were incredibly expensive and valuable. And all of a sudden, within a matter of decades, you had vanilla extract being mass produced incredibly cheaply and distributed all over the world. And this really showed people that there was a market for these flavors and that they could, in fact, provide very effective economics. So after this vanillin story, scientists started looking into other flavors, other fruits, and commodities that they could do the same thing with. And indeed, they were successful. So in the late 19th and early 20th century, you have a string of fruit flavors suddenly becoming available. Some of the most popular were butter flavor, which ended up being added to regular butter so that they can make it taste more buttery, which was kind of interesting. And then there was also the fruit flavors that were most popular were apple, grape, and strawberry. And I have a couple of examples on the slide of the specific compounds that are causing those flavors. But since there was this new burgeoning industry, in order to have some regulation of it, the FDA decided to make a distinction. So natural flavors are defined as flavoral chemicals that have been extracted from naturally occurring plants, animals, and yeast, whereas artificial flavors are flavor chemicals that have been extracted from anything else. So interestingly, these are often giving you the exact same compounds. So it's based on whether you extract a chemical from an apple or you make it in a test tube. That's the determination that decides whether it's natural or artificial. It doesn't matter what the chemical itself is. So how do they work? This is the complicated chemistry slide that I will try to break down and make as simple as possible. Almost all of the most popular flavor additives are aromatic esters, and this makes sense because we know that our more specific sense for determining what something is olfaction, our sense of smell, and esters are relatively small, easily carried by the air, and very strong smells. So for that reason, we have to get into how we can mass produce esters because they're not super common. And it turns out that the Fischer esterification process is the best way to mass produce these at a large scale. And it requires reacting a carboxylic acid with an alcohol, and another acid, usually sulfuric acid, is used as the catalyst. So let's look at the diagram here of the reaction mechanism. You start off with your carboxylic acid group, obviously, but because it's an acidic medium and there are free H plus ions, it's going to form a coordinate covalent bond with that top oxygen's lone pair. And what that gives you is this sort of resonant structure, sort of, since you have the extra electrons now traveling between the oxygens and the carbon. And that's why you have like 3 different forms of it in brackets at the top. Now, once you've formed that and you've made this carboxylic acid more reactive, it's going to be attacked by what we call neutrophilic alcohol. This is an alcohol that wants to get rid of its electrons, and there's this partial positive charge on the carboxylic group, so it'll attack it and try and release its electrons there. And now you have a very unstable activated complex on the carboxylic group because it has 2 hydroxyls attached to it. And then at the bottom, it's attached to the oxygen that's attached to the rest of its chain when it combines with another alcohol. Now, with the release of H+, since this thing's stable, something has to give. So you're going to end up releasing a hydrogen ion, and now the structure needs to be realigned. And now, again, this still doesn't work because the oxygen has 3 bonds instead of 2. So it's what we call an oxonium ion that needs, or not an oxonium ion, but it's what we call an oxonium complex that actually needs to be released. So that will come off in the form of water, and now you're left with the 1 oxygen that you need and the second one in the R group. So you can see in the final step there that we have the classic ester structure of a carbon bonded to an oxygen in the chain, and there's another double bond O on the carbon. And producing this turned out to be relatively cheap and relatively efficient because all the necessary ingredients could be determined. So if you decide what specific carboxylic acid you need and what specific alcohol you need, then you can make this very, very effectively and very reliably. So for that reason, this became the dominant method of producing esters. Fischer esterification became very effective. But that begs the question, what do these esters really do in our body, right? Because we're going to be eating them, and the primary mechanism by which we can get rid of them is through urination. Since these artificial flavor esters are usually the same as the ones you'd find in the original fruit, they're not so different in how we handle them. They can be absorbed in the small intestine, just like most alcohols are, and then they're sent by the portal vein to the liver, where they can be modified by a process called glucuronidation, where you attach an acid group on them to make them more water soluble. After the modification, the molecules can enter the bloodstream because we know that as soon as they come around and reach the kidney, a nephron will filter them out and they can enter the urine. The secondary mechanism by which we get rid of them, of course, is if they're never absorbed in the first place. And in that case, they just continue to travel through the colon and the stool. So let's do a case study of artificial sweeteners, the most specifically popular type of artificial additive today. And let's make something very clear. These are different from the esters we just talked about. These aren't artificial flavors because these are molecules that are directly acting on the taste bud receptors in the tongue. And how do we make those? Well, we find something that can essentially disguise itself with glucose-like residues that are on it and create a group that can react with the proteins that glucose reacts with in the same way. So the purpose of these, they're very useful. They have three big advantages that they offer. The first being that they assist in weight loss because it forces the body to burn fat for energy. The second being that it increases the shelf life of products and also hugely decreases the production cost because these are very cheap to make. And thirdly, it isn't as harmful to the teeth for the consumer because the bacteria in our mouth don't fertilize it the same way, or not ferment it the same way. So the common types of artificial sweeteners, there's 7 that are legal in the United States, but these 3 kind of have a monopoly on the business. The first is stevia, which is plant-based, and it's made from glycosides, which are these chains. It's a chain of 3 rings with these glucose residues that are on it, and the glucose residues essentially let it mimic glucose because it looks very similar and it can bond to the receptors in the same way. However, it isn't digested the same way as glucose is. We can't break it down as easily. And so what happens is that's why it assists in weight loss, because we're eating stevia instead of sugar. The stevia isn't something we can extract energy from, so we start burning muscle or fat instead. Next is aspartame, which is a methyl ester of aspartic acid. This is like coincidentally an ester. It's not really aromatic. It's too large. But this binds a G protein receptor, and it does it much the same way because of the aspartic acid and the methyl group. It can mimic glucose with its structure. And then thirdly, we have sucralose, which is selective chlorination of sucrose. So this is taking sucrose and replacing the hydroxyl group, or replacing certain hydroxyl groups with chlorine ions. And that makes it taste the same way in our mouth, but again, be digested a completely different way in our stomach. In fact, it can't be digested at all. We just don't do anything with it. Sucralose is the one in Splenda, by the way. It's the most popular, and the reason for that is because it's been shown to be not carcinogenic, while the other 2, there have been some studies that suggest that they might be somewhat carcinogenic. There's not enough information on that, and there's too much controversy for me to really get into it further. So final notes, there are some non-ester flavor additives. I only talked about the esters here, but there can be others, of course. But they're still going to be very similar because they have to be these small carbonic molecules capable of being carried by the air. So even if they're not esters, they're going to look more or less similar to the esters, and they can be ketones sometimes. Second of all, there's still controversy about how growing use of GMOs is going to affect natural versus artificial flavor classifications. This one's interesting because, like I said, the flavor classifications only care about where you get the chemicals. They don't care what the chemical is. And in this case, people are questioning whether GMOs count, whether, because if I extract chemicals from a strawberry and use them in my strawberry flavor, that counts as natural flavor. So they're essentially asking, what if you extracted it from a genetically modified strawberry? And there's no easy way to answer that sort of satisfies everyone. And people are proposing that soon the FDA will have to get rid of natural versus artificial flavor entirely and come up with a more detailed, more rigorous way of differentiating between the different types. Next of all, we have a fun fact that flavor is actually a really competitive industry. So it was kind of hard finding any specific examples of how they make a specific flavor additive because they're so secretive about it. So the Fischer esterification example I showed you is like the most general type. But if I tried to show you the Fischer esterification for the formation of vanillin specifically, for example, like people have their secret methods of making it that they don't want other companies to know. Also, a fun fact that about 60% of the artificial flavor factories, there's just a row of 7 factories in a row off of the New Jersey Turnpike, which is like most of America's flavor industry. So if you're interested in a field trip, that might be a fun place to go. And lastly, you know what? All this talk about food and taste and health, remember to eat smart. There is no substitute for good health. With that in mind, here are my sources. Thank you so much for listening to my presentation. I hope you have a excellent rest of your class, and just look to the person next to you and reflect on how glorious chemistry is. That's your activity for the rest of the period. All right, that's all.